Can biotin help detoxify oxalate? This is a hypothesis that may reconcile some anecdotes and has many hints suggested in the enzymological literature. Sally Norton, in her new book Toxic Superfoods: How Oxalate is Making You Sick, brought it to my attention that oxalate inhibits several enzymes, including the biotin-dependent enzyme pyruvate carboxylase. In this episode, I will go a step further. I will propose the hypothesis that the biotin-dependent enzyme pyruvate carboxylase actually detoxifies oxalate. This point that Norton raised struck me as very, very relevant to my own investigation of what, at the time, I believed was primarily a biotin-dependent issue, but I now believe is a mix of poor biotin recycling, poor ability to add biotin to biotin-dependent enzymes, and a shift of biotin away from pyruvate carboxylase toward a different enzyme, propionyl-CoA carboxylase, that is also biotin dependent, all based on genetics, combined with some other things that I'll talk about in a future episode that are also genetically driven. But at the time that I read Norton's book, I was investigating the possibility of a very biotin-centric hypothesis for my own stuff. And one reason it struck me as very relevant is that I know for a fact that I have, or at least have had in the past, issues with oxalate. Most glaringly, roughly 2 decades ago, I discovered how amazing sweet potato fries were, so I bought a bunch of sweet potatoes. I ate some of them as fries, some of them baked, averaging about 1 to 2 sweet potatoes per day, and by the end of the week, I had a limp. My emerging neck tension at that time had also increased considerably during that week. I said to myself, I must be eating something high in oxalate. I looked it up, and sure enough, sweet potatoes were up there with spinach. So I cut them out, and within a week, the limp was gone. Could I be confusing oxalate toxicity with biotin deficiency? Not entirely, since my concern about biotin is that my biotin levels are discrepantly low compared to my diet, and I have found as a fact that I have genetic variations that are rare that I just told you about, that all combine to create a situation of lower than expected biotin status with lower than expected pyruvate carboxylase status relative even to the low biotin status. So I know that that issue exists. Now, could biotin deficiency be making me more vulnerable to oxalate? That would explain why my mother has a need for high-dose biotin for her hair health and is vulnerable to oxalate kidney stones, which I think reflects a gene we inherited from my maternal grandfather, who had severe hearing loss rather early in life. This also seemed very relevant to numerous discussions brought up in the comments on my biotin posts, especially when high-dose biotin is truly needed. In the comments on that post, Jen P. wrote that 2 of her children were supplementing with biotin to help with yeast and oxalate intolerance. They did well on 5 milligrams, but 10 milligrams made their hands break out in terrible rashes that took months to heal. Similarly, Caddy Girlyo asked me on Twitter why 2 milligrams of biotin gives her excess keratinization of the skin. Could these represent what Susan Owens calls oxalate dumping? Norton singles biotin out as one of the 3 B vitamins of special concern since oxalate hurts biotin-dependent enzymes. In table 15.6 of her book, she lists biotin in doses ranging from 0.5 to 10 milligrams per day to help reduce dumping side effects, including brain symptoms. That's a quote, reducing dumping side effects, including brain symptoms. I'm guessing that this comes from Owens' work. On Owens' website, she notes that oxalate produces symptoms similar to biotinidase deficiency, which is a genetic deficiency in the ability to recycle biotin. Since one of these is runaway yeast infections, she suggests, quote, a supplement that furnishes biotin in milligrams rather than micrograms to help with chronic yeast issues, end quote. She also lists, quote, strange rashes, end quote, as a sign of dumping. Norton prefers the word clearance to dumping, but embraces the idea that low oxalate diets can reduce circulating oxalate down to a threshold that favors oxalate clearance over oxalate accumulation and can lead to such symptoms. These can include any symptoms of oxalate toxicity, and she lists eczema as one in table 10.1. In the beginning of chapter 9, she lists among the symptoms of clearance gritty snot, fine grit, white crystals or white dust coming out of the skin and nail beds, skin peeling off the fingers and toes, red or white bumps on the skin, and terrible rashes. I am generally skeptical of detox reactions. I believe they exist, but I also believe they are often used to set up unfalsifiable defenses of protocols that don't work. However, it makes complete sense to me that reducing circulating oxalate beneath a certain threshold will stimulate the clearance of accumulated crystals. Formation of crystals would be a defense against biochemical toxicity, but at the expense of damaging connective tissues and mucous membranes. Reversing this process could temporarily increase biochemical toxicity. Moreover, when crystals break up, they never fully solubilize automatically, so the breaking up of crystals could cause physical irritation of tissues. Neither Owens, to my knowledge, nor Norton suggest that biotin could cause clearance reactions. Yet Jen P. and Caddy Gurlio report experiences that could be interpreted in that light. Jen P. was particularly confused by the reaction. She says, quote, "I was told later that it probably pushed out oxalate too fast for their bodies to handle, thus the rash. This has never totally made sense to me, as biotin is meant to help the body eliminate oxalates and should be correcting processes that oxalate has disrupted." End quote. Evidently, someone is saying that biotin can promote clearance of oxalates. Can it? This is only a hypothesis, but as I looked into the enzymological literature, I became more and more convinced that the biotin-dependent enzyme pyruvate carboxylase is a plausible candidate for a means of detoxifying oxalate to formate, which can be utilized in methylation or further broken down to carbon dioxide. First, let's take a look at what oxalate is. Before we get going, I just want to let you know that I have a free gift for you. That is my MTHFR Protocol, a 7-page quick guide to optimizing and personalizing your methylation status using foods and supplements. My MTHFR Protocol is free to anyone who subscribes to my Substack, whether free or paid. Use chrismasterjohnphd.com/MTHFR or use the first link in the description. Enter your email, click subscribe, and you will immediately get a welcome email in your inbox that contains my MTHFR Protocol as a downloadable PDF. I hope you find your free gift useful and enjoy it. Once again, that's chrismasterjohnphd.com/MTHFR. My hypothesis is that oxalate can be broken down to formate and carbon dioxide. So take a look at the structure of oxalate, and you will see that oxalate is simply 2 carbon dioxide molecules bound together. This is the simplest dicarboxylic acid. It's just 2 carboxyl groups, because all a carboxyl group is in organic chemistry or biochemistry is what you get when you add carbon dioxide to an organic molecule. So this is just 2 carbon dioxide molecules stuck together to make one molecule with 2 carboxyl groups. That's all that it is. And so something seems very strange about the idea that we cannot break this down. After all, what do we do with everything we eat? We break it down to carbon dioxide and whatever else we can take out of it. And so it would be crazy if this little measly thing was the most toxic thing on the planet. We could do nothing about it except damage our joints as a defense against the biochemical havoc that it would wreak in our system running freely. There should be a way to just split this mother in half and blow it out in the breath. That's there. We should be able to do that. Now, take a look at the structure of formate. And formate is simply carbon dioxide with 1 hydrogen sticking off of it. That's all it is. So formate is almost carbon dioxide. So keep that in mind as I go through the details of my hypothesis here. This thing is in any diet on the earth at tens or hundreds of milligrams per day, has toxic effects at physiological concentrations, and we are supposed to not be able to split it into 2 molecules of CO2. The only way we can break this thing down is to hope our gut bacteria, contain the rare organisms that specialize in feeding off of it. That is the conventional belief. So what do I think? Well, before I talk about how I think that humans should be able to break down oxalate, let's look at what is known about the detoxification of oxalate in bacteria. This is not comprehensive, it's just illustrative to get a glance of what oxalate detoxification should look like if it indeed occurs in humans. So, to illustrate what it often looks like, several Pseudomonas species metabolize oxalate. Pseudomonas oxalaticus uses succinyl-CoA to donate the CoA to oxalate, forming oxalyl-CoA. It then reduces oxalyl-CoA to glyoxylate and coenzyme A, or CoA, using NADPH. Thus, pantothenic acid or vitamin B5 is used in the form of coenzyme A, or CoA, and niacin or vitamin B3 is used in the form of NADPH. To use oxalate for anabolic reactions, the bacterium feeds glyoxylate into the glycerate pathway. To catabolize oxalate for energy, oxalyl-CoA is decarboxylated to formyl-CoA and CO2. Formyl-CoA is just formate that's bound to coenzyme A. The CoA that is released is transferred to a new oxalate molecule, while NAD+ is used to oxidize formate to CO2. There are several pink pigmented Pseudomonas organisms, known as AM1, AM2 ruber, and extorkins, that do the same thing, but have an alternative way of using oxalate for anabolic reactions. They make serine, an amino acid, react with glyoxylate to form hydroxypyruvate and glycine, while folate, vitamin B9, donates a carbon to glycine to regenerate serine. The main point here is that they all use the enzyme oxalyl-CoA decarboxylase to break off the first CO2, which makes formate, and then they use formate dehydrogenase to release the last CO2. All it does is take that extra hydrogen off formate, because remember, oxalate is two carbon dioxides glued together. Formate is just CO2 with a hydrogen atom glued onto it. So oxalyl-CoA decarboxylase breaks off one CO2, blow it out. Of course, these are bacteria, so they don't blow, but release it into the air, into the solution in the cell that can then be released into the air later. And then what you have left is you have formate, because you have a hydrogen that was part of what was gluing these bonds together, or reflecting the extra electrons that were essentially gluing these bonds together. And you use formate dehydrogenase to pull it off. You pull that hydrogen off formate. What do you have? You have CO2. Now you have the second CO2. Release it, right? So if bacteria want to destroy oxalate, they need to oxidize it and rip it apart to 2CO2 and release the 2CO2. The question is, can humans rip oxalate into 2CO2, oxidize it, and rip it apart into 2CO2 and release it? So before we get to the controversial part, let's look at human metabolism, what is not controversial. And so that is human formate metabolism. Okay, so what are humans known to do with formate? Because this all is about the second step of oxalate detoxification. So formate can be formed from serine, glycine, methionine, choline, and methanol, and may also be formed during the synthesis of cholesterol and other sterols and the breakdown of tryptophan. Gut bacteria can make formate and methanol, and exposure to formaldehyde or methanol in industry or laboratory work are additional sources. An overflow of formate is a major cause of methanol toxicity, but we have excellent ways of putting formate to good use or detoxifying it when we are not exposed to excessive or surprise exogenous doses. Since formate possesses one carbon, it can provide the one carbon for so-called one-carbon metabolism, also known as methylation. The principal disposal route for formate is to provide the carbon that becomes the methyl group of methylfolate. Interesting. Note here that this is a tangent, but for all the people who think that the world is split into two groups of chemicals that are toxic and chemicals that are good for you, consider that formate is the primary route of methanol toxicity and is also one of the primary molecules that becomes the methyl group of methylfolate. Because the world is not divided between two categories of things, good things and evil things. It is rather full of good things that can be used well to contribute to health or can be used horribly to contribute to disease. I realize it gets complicated when you talk about outright poisons, but this is a tangent. Okay. Going back to the main story. If we add more formate to folate than is needed for methylation, we use the niacin-dependent enzymes aldehyde dehydrogenase 1 and 2 to break the formate off of folate in the form of CO2, releasing it while we exhale. So, one thing is definitely true. Like bacteria, we, humans, can use niacin to oxidize formate to CO2. Thus, we clearly possess the second expected step of oxalate detoxification. All right? The only question is whether we have a way to decarboxylate oxalate to form formate in the first place. That is, do we not only possess the second step of oxalate detoxification, which is to convert formate to CO2, do we possess the first step to decarboxylate oxalate? And this is where it gets controversial. And this is where what I'm saying here is uniquely my hypothesis. I don't know that no one else has proposed it before, but to my knowledge, I'm proposing it originally. I'm sorry to interrupt you, but it's only going to be a second. I just want to remind you that I have a free gift for you. That is my MTHFR Protocol, a 7-page quick guide to optimizing and personalizing your methylation status using foods and supplements. My MTHFR Protocol is free to anyone who subscribes to my Substack, whether free or paid. Use chrismasterjohnphd.com/MTHFR or use the first link in the description. Enter your email, click subscribe, and you will immediately get a welcome email in your inbox that contains my MTHFR protocol as a downloadable PDF. I hope you enjoy your free gift and find it useful. Once again, that's chrismasterjohnphd.com/MTHFR. And with that, back to the episode. Okay, pyruvate carboxylase is known to be a multi-substrate enzyme. The textbook function of pyruvate carboxylase is to allow glucose to enter the citric acid cycle using itself as its own fuel, and to allow protein or lactate to be used to synthesize glucose. Glucose entering the citric acid cycle is needed for it to be burned for energy, and is also needed for it to be converted to fat. As a result of this, pyruvate carboxylase is needed indirectly to synthesize anything fatty, such as the elongated forms of the essential fatty acids, the myelin sheath of neurons, or lipoic acid, which is a modified fatty acid that acts as a cofactor for several enzymes. Specifically, pyruvate carboxylase does all these things by taking CO2 that has been dissolved in water to form bicarbonate and adding it to pyruvate to form oxaloacetate. The biochemical adage, "fat burns in the flame of carbohydrate," refers to the fact that fatty acids generate the 2-carbon acetyl-CoA but cannot lead to the net synthesis of the 4-carbon oxaloacetate. As a result, the acetyl-CoA they release cannot be fully burned for energy and condenses with itself to form ketone bodies, some of which leave in the urine. However, in the presence of glucose, pyruvate carboxylase can convert pyruvate derived from glucose to oxaloacetate to allow the acetyl-CoA to be burned for energy. During a ketogenic diet, as an example, or fasting, oxaloacetate is used in the liver for gluconeogenesis, and incoming pyruvate is in short supply. This depletes the liver of oxaloacetate. Acetyl-CoA from fatty acid oxidation accumulates. There is no oxaloacetate to allow the acetyl-CoA to enter the citric acid cycle, so the acetyl-CoA molecules condense with each other to form ketones. Glucose taken up by the brain, muscle, and fat is used to make pyruvate, and then pyruvate carboxylase converts the pyruvate to oxaloacetate, and this allows the ketone bodies made by the liver to be used for energy in those tissues. However, protein can enter the citric acid cycle by other means to produce oxaloacetate, and this is why, in my article, High Protein, You Need More Biotin, I suggested that one sign of biotin deficiency would be hyperglycemia that is mitigated by dietary protein. The adage should be, "fat burns in the flame of oxaloacetate." Despite this singular textbook explanation for the enzyme, in vitro, in a lab dish, the enzyme can easily run backwards to decarboxylate oxaloacetate. As one group wrote in 1974, pyruvate carboxylase is a multi-substrate, multi-effector enzyme. In vivo, in the live human or animal, pyruvate carboxylase does not run backwards, but this is only because the concentration of oxaloacetate is always vanishingly small. This is why urinary organic acid tests do not even bother measuring it. Any oxaloacetate that is not used to fuel the citric acid cycle or be converted to aspartate using the vitamin B6 dependent enzyme will be rapidly converted to malate, which is the backwards reaction of the last step of the citric acid cycle. Even earlier, in 1963, researchers had shown that pyruvate carboxylase can carboxylate other molecules. Specifically, it carboxylated alpha-ketobutyrate, a product of cysteine metabolism, at 3% of the rate at which it carboxylated pyruvate. Thus, pyruvate carboxylase is at least capable of running backwards to perform decarboxylation and of metabolizing substrates other than pyruvate and oxaloacetate. Okay, so the first thing that I'm establishing is that it is known, number one, that pyruvate carboxylase can run backwards to decarboxylate things, and number two, that pyruvate carboxylase is known to metabolize other substrates other than the pyruvate to oxaloacetate interconversion. So it can run backwards, it can metabolize other substrates than are what in the textbook. Okay, now let's take a look at the literature showing oxalate inhibits pyruvate carboxylase. I'm going to put inhibits in quotes, and I'll show you why in a second. So the interest in the ability of oxalate to inhibit pyruvate carboxylase goes back at least to the 1960s, when numerous authors showed that oxalate inhibits this enzyme in baker's yeast, bacteria, and rat liver. In the 1980s, research picked up on the physiological relevance of this inhibition. In chicks, oxalate was shown to inhibit gluconeogenesis and also to inhibit cholesterol synthesis and the conversion of lactate into fatty acids. As noted in a paper from 1983, oxalate is present in human plasma at concentrations between 20 to 40 micromoles per liter. At these concentrations, experiments suggest it should freely enter liver and muscle cells, where it would inhibit pyruvate carboxylase along with at least two other enzymes of pyruvate metabolism. This paper showed that in rat liver cells, physiologically relevant concentrations of oxalate inhibit the citric acid cycle activity by a whopping 48%. As would be expected, this leads to the reduction in the amount of mitochondrial NADH, the opposite of what a respiratory chain disorder would do, and to increased ketogenesis as a result of decreased oxaloacetate. The reason I put inhibits in quotation marks is that not one of these studies, indeed no study that I can find anywhere, looked at whether oxalate was being decarboxylated to formate while it was inhibiting pyruvate carboxylase. In other words, it is inhibiting the reaction you want to happen. But is it only staying stuck in the enzyme and blocking the pathway? Or is it using the enzyme for the first expected step in its own detoxification, its decarboxylation to formate? Can pyruvate carboxylase convert oxalate to formate? So what we've established so far is pyruvate carboxylase can run backwards to decarboxylate. It can metabolize other things. It does interact with oxalate, with the established papers showing that oxalate inhibits it. None of those papers look at whether oxalate, during its inhibition of the conversion of pyruvate to oxalate, is actually becoming decarboxylated by the enzyme to formate. Because if that's true, that would mean that we possess the first step of oxalate detoxification expected from bacterial metabolism, and it is already known we possess the second step. Okay, so can pyruvate carboxylase convert oxalate to formate? How plausible is this? Using pyruvate carboxylase isolated from chicken liver mitochondria, one group showed in 1966 that oxalate inhibited the forward reaction in a non-competitive manner, but inhibited the backward reaction, which is the decarboxylation of oxaloacetate, in a competitive manner. What that means is that oxalate was binding somewhere in the enzyme other than where pyruvate binds, but in effectively the same place where oxaloacetate binds during the backward reaction. So oxalate mimics oxaloacetate with respect to its where it's situated in the enzyme during the decarboxylation of oxaloacetate. Fast forward a half century to a paper from 2013, and we learn much more. Number one, oxalate binds to the active site of the enzyme 15 times more strongly than pyruvate does. Number two, oxalate stereochemistry, which refers to the orientation of its molecular orbitals, essentially the geometric conformation of its electric charge, looks just like that of enol pyruvate, which is an intermediate in both the carboxylation of pyruvate and the decarboxylation of oxaloacetate. Number three, oxalate tucks one of its carboxyl groups right up against threonine 882, the specific amino acid that donates a proton to enol pyruvate during the decarboxylation of oxaloacetate. Pair this with what we knew from the studies I reviewed above, that pyruvate carboxylase can run backwards, that pyruvate carboxylase can metabolize other substrates such as alpha-ketobutyrate. Everything about this is screaming that pyruvate carboxylase can decarboxylate oxalate to form formate. Yet I've not found one paper that so much as looked for this. So what we've established is pyruvate carboxylase can decarboxylate by running backwards. Pyruvate carboxylase can metabolize other known substrates. Pyruvate carboxylase not only reacts with oxalate, but oxalate tucks itself into exactly right down to the specific amino acid in the active site, exactly where you would expect it to be if it were decarboxylated to formate by the enzyme. Okay, suggestions from other enzymes. The bacterium Klebsiella aerogenes possesses a biotin-dependent oxaloacetate decarboxylase. The enzyme binds oxalate 30 times more strongly than oxaloacetate. The authors suggested, quote, decarboxylation of oxaloacetate probably goes through an enolate transition state, which would be perfectly resembled by oxalate and would thus explain its strong inhibitory power, end quote. So it would be perfectly resembled by oxalate, but oxalate would not be decarboxylated. They didn't address that issue explicitly, but I would think that perfect resemblance of the transition state would lead to similarly perfect resemblance of the actual reaction. More recently, in a 2018 paper, the authors attempted to explain the bifunctionality of a, quote, human oxaloacetate decarboxylase known as fumarate acetoacetate hydrolase. This enzyme will cleave fumarate pyruvate into fumarate and pyruvate, and will cleave acetopyruvate into acetate and pyruvate, yet will also decarboxylate oxaloacetate to form pyruvate. The authors did not investigate whether oxalate could be decarboxylated to formate, because they were looking at the crystal structure of bound substrates and inhibitors. They noted in the crystal structure that oxalate forms, quote, a potential catalytic triad, end quote, which means that it orients itself in the enzyme in a way that might lead to its catalysis, that is, its decarboxylation to formate. Thus, there is a general trend that oxaloacetate metabolizing enzymes bind oxalate even more strongly and in a way that suggests that oxalate would be decarboxylated to formate. So what have we established now? Pyruvate carboxylase can run backwards to decarboxylate. Pyruvate carboxylase can metabolize other substrates. Pyruvate carboxylase interacts with oxalate in exactly down to the geometric conformation of the electric charge and the exact positioning within the active site in exactly what you would expect if oxalate were decarboxylated to formate. And whenever we can find other oxaloacetate metabolizing enzymes, the trend is that they all bind oxalate more strongly and in a way that you would expect would lead to the decarboxylation of oxalate to formate. Okay, now, would biotin be important? Although biotin is a cofactor for pyruvate carboxylase, it's not automatically obvious that biotin would be needed for its decarboxylation to oxalate. Biotin is an anabolic vitamin, and its role is to use the energy of magnesium ATP to add carbon dioxide to molecules. The way this happens in pyruvate carboxylase is as follows. Number one, biotin is carboxylated using magnesium ATP to form carboxybiotin. Number 2, carboxybiotin moves to a second location within the enzyme. Number 3, biotin liberates the CO2 to form biotin enolate. Number 4, biotin enolate takes a proton from a nearby threonine, which then takes a proton from pyruvate, forming enol pyruvate. Number 5, enol pyruvate is unstable and it attacks the CO2 that biotin is liberated. And finally, number 6, the joining of the CO2 to enol pyruvate forms oxaloacetate. When the enzyme runs in reverse, oxaloacetate takes a protein from the threonine rather than pyruvate giving it up. Energy is released rather than consumed. The energy is used to make magnesium come together with ADP and phosphate to form magnesium ATP. Biotin is needed to help invest energy into carboxylation. What role could it have in decarboxylation? Well, it might have one. Even if it's not doing its typical job, it's still part of the machinery. Even if you are driving downhill, your engine is still on, for example. If you had no car engine, you wouldn't be coasting downhill for very long. The 1974 paper found that avidin, which binds to biotin, does not completely inhibit the decarboxylation of oxaloacetate in the way that it does completely inhibit the carboxylation of pyruvate, but it does double the amount of oxaloacetate needed to get the same rate of decarboxylation. In other words, an absence of biotin would not destroy your ability to decarboxylate oxalate, but it would double the amount of oxalate needed in circulation to get the same amount of detoxification. So if I'm right about this, biotin would promote oxalate clearance. If it promotes oxalate clearance, it could perhaps lead to temporary symptoms of what Owens calls dumping. The bottom line. It has never been demonstrated that pyruvate carboxylase can decarboxylate oxalate. However, numerous lines of circumstantial evidence suggest that it is likely to participate in the first step of oxalate detoxification, its conversion to formate. This would allow formate to enter the methylation cycle or be converted to CO2. Together, this would allow oxalate to be broken down into 2 molecules of CO2 and exhaled. If this is correct, biotin could help promote oxalate clearance. This may suggest the need to go slow in some cases where rapid clearance would lead to temporary bouts of uncomfortable symptoms. This is over and above what Owens and Norton note, which is more clearly documented, that oxalate inhibits biotin-dependent enzymes at physiologically relevant concentrations. Oxalate is a metabolic toxin. The fact that Owens and Norton list milligram doses of biotin as helpful for clearance symptoms suggests to me that it is usually more relevant that oxalate is hurting the normal textbook functions of the biotin-dependent enzymes than it is that biotin provokes more rapid clearance, and as a result, it is more often helpful to have higher doses of biotin during this period of clearance than it is to experience adverse effects from them. However, I would note that it may simply be more often relevant that it is clearing oxalate, and yet that is helping rather than hurting, because it's not necessarily the case that whenever you promote the clearance of oxalate, you are always causing dumping symptoms. So I do think it's possible that if this is true, that the normal plasma and urine responses you see to oxalate from the diet are not at all a complete picture of the oxalate that you absorbed, but are rather reflecting the oxalate that you absorbed but didn't detoxify because you detoxified most of it. In any case, my hypothesis leads to 2 important insights. Number 1, biotin deficiency may directly lead to oxalate intolerance. And number 2, in the occasions where biotin supplements lead to skin rashes, it may not always be about balance with other nutrients, but it could perhaps be direct supporting of more rapid oxalate clearance and thus dumping symptoms. All right, so I don't have any action strategies to take directly out of this because it's just a hypothesis. However, you may choose, if you have oxalate related issues, you may choose to use this hypothesis in a way that could inform how you interpret your experience in the action strategies that you do have, including the fact that if you are taking biotin, it may help with the clearance of oxalate, which may be more often than not beneficial. But if you use too high of a dose and do get oxalate-related symptoms to it, that may be because the biotin is helping to clear the oxalate. Mostly, I hope that that scientific researchers in the enzymological field will look at whether pyruvate carboxylase can indeed convert oxalate to formate, because it's obviously the case that that some scientists are set up to look at exactly these things. I have more on biotin. In particular, you may want to see my articles, High Protein, You Need More Biotin. Biotin causes a multitude of false lab tests and when high-dose biotin is truly needed, on my Substack. And my simple food-based recommendations for getting enough biotin are found in my Cliff Notes, Vitamins and Minerals 101 Cliff Notes. And if you want the references, then head to my Substack. I'll put the link in the description. I would also recommend heading over to that link just to view the comments. There are a lot of great comments there, including one by Susan Owens who stopped by. And I'll just read this one comment before I go to elicit your potential interest in heading over to the comments there and participating. So Susan Owens wrote in response to this post, hi Chris, when I first began to obtain the background I needed to understand the oxalate issues, I found in autism 18 years ago in 2005, I attended professional oxalate conferences. I was very surprised to discover they were only attended by kidney doctors and kidney researchers. To them, finding oxalate elevated in autism was an anomaly. In fact, finding oxalate elevated apart from kidney stone disease or genetic hyperoxaluria were in their minds impossible. Why did I start studying it? Beginning about 30 years ago, my academic work was all about sulfate and discovering its role in neurodevelopment. Years later, sulfate was found to exchange for oxalate on a special set of transporters called the SLC26A family of transporters. I had always thoroughly explored the role of sulfate in autism, but this new science meant that now I needed to study oxalate to find out how oxalate could disrupt sulfate and other things important in autism. To accomplish that, I studied the oxalate literature for 4 months with a special focus on the genetic hyperoxalurias, where it was known that oxalate traveled to the whole body. I attended 7 professional oxalate conferences. To my surprise, the scientists there seemed to have formed an unspoken pact to ignore issues that were not centered around kidney stone disease. They were uninterested in how oxalate affected the general biochemistry wherever it went. I had already spent a decade recognizing complex issues in autism, so I knew that oxalate, by affecting sulfate, could alter neurodevelopment as well as hormone and neurotransmitter regulation, and it was a clear mitochondrial toxin. This new information required a reinterpretation of autism findings. Before that, I had been studying sulfate's role, but now oxalate could be a major disruptor. Formate was also a substrate of those transporters. Anyone can find literature about how formate and oxalate have been influencing each other, but I found nothing practically useful. Chris, this is why I'm particularly delighted to find your interest in this field, and I like the way you were looking at it, in the way it should have been studied a long time ago. The oxalate field for so many years has blinders on, something that Sally Norton and I have both found and explored independently. Sally has been more into thinking about issues with diet, but I became more concerned about reaching academia and helping them study the oxalate the body makes under stress. It was impossible to do a study in the US on oxalate in autism because the labs in the US that were skilled in measuring oxalate told me they would not test anyone unless they had kidney disease. That is why I recruited a highly qualified group of scientists to look at oxalate levels in autism, and we got astonishing results. That paper is ranked now in the top 4% of scientific studies, and it has had 42 citations. That attention is really great, but it is still not motivating other scientists to look past the kidneys and find other types of diseases related to oxalate. Chris, the commendable sort of thinking that you are doing is exactly what is needed to move oxalate research forward and into new territory. I eagerly invite you to further explore this fruitful area that has been in sore need of your skill set. I hope that you can generate experimental evidence. In 2005, with a bunch of grateful autism parents, we formed the Trying Low Oxalates Network, and it has already helped more than 70,000 people discover the relevance of oxalate to their own health. This is why we picked up an enormous amount of personal experience about reducing oxalate, but we've not seen nearly the amount of change we want to see in academia. We're trying to change that. One group performed the most successful fundraising of anybody this past year for the Oxalate and Hyperoxaluria Foundation for Giving Tuesday, and that is because our vision is to support the sort of thinking you are doing to move the study of oxalate into a new era, where multidisciplinary and cross-disciplinary work rules the roost instead of being barred from the roost. Kudos on your deep thinking. I would be delighted to see people like you get funded so that we can get together to solve the many human diseases related to oxalate. Best wishes, Susan Owens, head of the Autism Oxalate Project at the Autism Research Institute, founder of trying low oxalates network since 2005. All right. Well, I read that mainly so you can head over to see the other 57 comments that are on the article and maybe leave your own. And with that, we'll see you in the next episode. Before you go, I just want to remind you one more time that I have a free gift for you. That is my MTHFR Protocol, a 7-page quick guide to optimizing and personalizing your methylation status using foods and supplements. My MTHFR Protocol is free to anyone who subscribes to my Substack, whether free or paid. Use chrismasterjohnphd.com/MTHFR or use the first link in the description. Enter your email, click subscribe, and you will immediately get a welcome email in your inbox that contains my MTHFR protocol as a downloadable PDF. I hope you enjoy your free gift and find it useful. Once again, that's chrismasterjohnphd.com/MTHFR. See you next time.